Good morning, everyone. It's really good to see so many familiar faces, and I met some new people too. I'm Sanjoy Dutta with JDRF in the research department, and we have been long-time partners with the Type 1 Diabetes Exchange. We saw it being born, grow, and be so successful as it is today. And so it's my privilege here to be presenting the work of our team in supporting research at JDRF here, and many of the investigators that we work with are also in this room. So I'm really in very, very good company. Let me see if I know how to make this work. I do. All right, I think most of you are familiar with JDRF. I'll go over some of our areas quickly, and then I'll focus a little more on what we call improving lives, because Osaki had asked me to emphasize that area a little more than some of our cures area, but I'm happy to take questions in any of the areas. And my colleague, Dr. Albanese O'Neill, is in the audience. She can also field questions as needed. You know, we have been in existence now for over 50 years with the mission of, changing lives of people with type 1 diabetes or at risk with type 1 diabetes. So cures, preventions, and treatments for T1D and its complications. How can we make people's lives better? But actually what we are secretly thriving for is a world without type 1 diabetes. So the next generation or the next person never sees type 1 diabetes. And we think that it is doable. It is doable given many of the advances we have had right now. So there have been so many advances, speaking of advances in type 1 diabetes, and if you ask me to rank order them, I probably will not be able to. I will not be able to rank order, but if you hold a gun to my head, this is New York, I can say that, then I would say probably staging type 1 diabetes has been a seminal contribution of people around the world, scientists, clinicians, many others, and we are glad to have been part of it from day one as a funder, as well as there have been other funders here. Staging has not only has led to so many things, a deeper understanding of type 1 diabetes pathogenesis. Who gets it? When do they get it? How fast do they progress? Who doesn't get it? But I think staging has opened the doors for now able to intervene before one goes on insulin. And that's a very critical part. And so I'll go over this quickly because I believe this audience is very familiar with the stages of type 1 diabetes. So as you all, again, as you are familiar, stage 3 is when one person is put on insulin because their body is no longer making sufficient insulin to be able to control their glucose levels or balance their meals or anything else. However, we now know that diabetes starts, or type 1 diabetes starts weeks, years, maybe even decades before you're in stage 3. In fact, yesterday at the JDRF New York Promise Ball, and every meeting, every event I go to, there was a young lady sitting next to me who I've known for a very long time. She was 12 years double autoantibody positive. One of her 3 other siblings has type 1 diabetes, so she's in the high-risk category. She converted in 2020 to becoming insulin dependent. And her mom had called me up, and teplizumab TN10 results had just come out, but teplizumab wasn't T-zealed. It wasn't yet a therapy in a vial. And so they were trying to get it, but they couldn't get it for regulatory and for other reasons. And of course, in hindsight, she wishes she had gotten it. Maybe she would have been able to delay it by 2.7 years or so. So that's what I mean is the power of staging beyond the knowledge aspect of it. These are some numbers that are very, very critical. They have not come out of the woodwork. They've come out of significant amount of research to suggest, at least in pediatric onset disease, that having 2 or more autoantibodies is just a matter of when, not if, that you become insulin dependent. And your lifetime risk is essentially 100%. This slide has been shown in many different formats by many different investigators. George Eisenberg has been credited, is probably the first person to have done this, a seminal work, and you can see this as a graph, as a slope, I've seen even a pie chart here, but this is how we have represented it here. Based on this staging paradigm, we have set priorities. As any organization, as any funding organization, we have to select priorities because there's always way more research to fund than we can. Based on that, in the top panel, you see our areas what we call are within our cures program, is if type 1 diabetes can be staged, if we can find out who is at risk, knowing that familial risk is higher, but about 90% or 85% of people that get it don't have a familiar or immediate family connection. Knowing all of that, screening for risk becomes a responsibility. It's not a choice anymore. It's a responsibility we all share. And Anastasia is leading a part of the implementation of that program here. And she's in the audience. Please do reach out to her with questions. Beyond screening, of course, we want to screen to know who is at risk. Knowledge is power. They can prevent decay or at least reduce the probability of decay significantly, have a smoother landing. All those are very, very important. However, our goal is to change the course of the disease. And screening provides us the tools to intervene in disease-modifying therapies, run clinical trials faster, and if you're eligible, probably avail treatments such as TZIELD, and hopefully many more to come in the years, that we see a lot of promise there. What about the 1.4 plus million people in the US and many more million people around the world that are already living with T1D, have a frank loss of insulin production, or complete loss of insulin production? What do we do for them? Yes, someday we can change the course of the disease, but we're not there yet. I don't see us going there in the immediate future. So for the people living with what we call established type 1 diabetes, sometimes loosely called stage 4 diabetes, cell replacement therapy is a viable option for what we call a functional cure, i.e. replace the lost cells with insulin-producing cells from outside the body, put it back into the body. So cell replacement has been a flagship program at JDRF for many years. I will not have time to go into details of either disease-modifying or cell therapies. I'll focus a lot on the bottom panel, that improving lives. Improving lives is literally what it means here. What can we do for people today while they wait for the curve to bend, to become insulin independent, to avail a cure? What do we do for them? How can we provide them good glucose control? How do we transition our mindset from a glucose-centric insulin monotherapy approach to a polypharmacology approach, considering metabolic control, considering prevention or delay in complications, and the huge importance of psychosocial and mental health that are frankly affecting people 24/7, 365 with this disease that requires such intense care and management here. And it's pretty unforgiving for mistakes. I don't need to say it to this room of clinicians and researchers who know this extremely well. One thing that we pride ourselves very much is not only the therapeutic pipeline that we are supporting here, or trying to accelerate here, but also the people pipeline. This is a field, unfortunately, in endocrinology that people are not chomping at the bit. So I am very appreciative of all the endos and other diabetes researchers in the room. We are very appreciative. We have a very robust training program with a pretty high rate of funding, over 20% year over year in the last 10 plus years here. We train postdoctoral fellows at different tiers of their postdoctoral training, all the way into establishing them as young faculties through career development awards. And so that has been important. And I know many of you in this room are recipients of that and many not in the room now. And so I encourage you all to look into our training program. Based on that, we'll just move into these cures areas very quickly. I'll focus most of my time, as I said, on improving lives. Screening is very important here. JDRF, Hempstead Charitable Trust, NIH, outside the United States, European Commission, others are all focusing on this because there is something tangible here one can do with either secondary prevention or even primary prevention here. Today, there's a lot of things we have to do. The state in which we are definitely is better than where we were 10 years ago, but we want to be in the blue area there. We want a world where preventive services are covered by payers in the US and outside the US. We have to be able to assess risk, identify these individuals, and intervene. This should not be a question of a choice. It's an onus. It's a responsibility we all share here. And I'm really proud to have worked at JDRF with so many other funders around the world, in the US and around the world here, that everyone is pulling in the same direction to get this one going. It is a huge lift. And we look upon you all to be able to support and help us get there. Just two more slides here. Why screening? I already mentioned here the importance of screening here. There are acute benefits. There are chronic, long-term benefits here. And so let's not lose sight of the fact it's not just preventing DKA at onset, but there's also a lot of other long-term benefits that come with it. And frankly, give people the choice if possible. With that in mind, we and others are funding several screening programs. These are the ones that JDRF is involved in. And you can see the importance of funding around the world because, one, we need a lot more data around the world around screening for knowing risk, assessing positive predictive value, how fast people are progressing, but also the heterogeneity of the population, the genetic heterogeneity, the environmental heterogeneity. There's a lot we need to learn. And pooling all the data sets together that we are involved in and others such as NIH, European Commission, Hampshire Charitable Trust, and many other funders involved in, will be able to provide the evidentiary basis to have healthcare systems actually cover screening and hopefully interventional therapies here. I'll quickly go with 2 slides in the disease-modifying therapy area, where the basic, the fundamental of this area of our area of focus here is changing the biology or the pathogenesis in type 1 diabetes. We know at the crux of it, there are at least 2 main biological issues in people with autoimmune diabetes here. One is the frank loss of beta cells, endogenous beta cells producing insulin. And then, of course, an active immune system, mostly T cell driven, but there's also B cell and other immune cell pathologies involved here. And there's a lot of debate on which comes first, but I frankly don't care. We got to fix both. With that in mind, we have several research programs in discovery and in clinical testing here, including, of course, our, sorry, let me just finish this. You'll see this pattern in many of my slides here, where we are today, but where we aspire to be in the future, where we are trying to drive the world along with others to the future. There is today we have one disease-modifying therapy that was approved almost exactly a year ago. It'll be a year tomorrow, on 17th of November. Last year, TZIELD got approved here. We are very, very proud of that, but it took 100 years since insulin was found. We can't wait that long. And how can we change that? How can we have multiple therapies in choice, just like we do with the device world, which I'll get into in a second here, and then have people not only have the choice, but also have the coverage to be able to change the course of the disease. This is the last slide in this area here about the approval of TZIELD here. Again, it took us 100 years to get another therapy out here after insulin. Of course, pramlintide has been approved in the US, but its uptick has been very low, as you all know here. This particular line of work, anti-CD3 being effective here, started in 1988, way back when. It took us 40 years to get here. Yes, the biology was not well known then. R&D takes time. It takes a lot of people to pool here, but we can't wait this long for the next new therapy to come along. There are several things we can do with the knowledge we have today and with increasing efficiencies around processes such as clinical trial. And I know this team, the Type 1 Exchange, can help us get there in a big, big way here. It took 10 years to do TN10. It shouldn't take 10 years. And there were challenges, but I'll say they did still a pretty good job. But let's try and figure out how we can be more efficient in screening for the high-risk individuals, in having clinical trial pathways that can get us to a decision much, much sooner than the trajectory here. But nonetheless, we're very happy that this was the icebreaker. And certainly you all know the left side of the slide here, that came out in 2018, the TN10 study here showing a delay in the onset of insulin dependence by a median of 2.7 years, initially 2 years, and then in the follow-up to 2.7 years. And then on the right, this is very, very fresh results coming out of the studies in stage 3 diabetes. So this is when you've been newly diagnosed, you've been put on insulin, showing statistically significant C-peptide, the surrogate of insulin production or beta cell health, compared to placebo. And this was presented by Dr. Harold from on behalf of the study team in Rotterdam at the ISPAD meeting here. There was a lot of other data sets presented there that trended in the direction of better overall glucometabolic regulation. They did not reach statistical significance, so we will have to see what the regulators think about this, given the results that's shown here. But nonetheless, it's encouraging, and we have a lot more to do and a lot more promise there. ADA was very prompt to, and in the middle of the year, which is not very usual there, they did change or enhance their guidelines, and we're very helpful. We're very proud of that because this allows clinicians to have a roadmap. This allows payers to anchor themselves in the guideline here. And so we are hoping that the whole field will now move at a pace that we haven't seen before, because research is moving quite fast here. As I said, I'll focus the rest of my 12 minutes time left here on the area of improving lives here. And this again is a very, very big area. The way we look at it here is based on the therapies. All of these are intended to improve the people's lives. Their clinical outcomes, clinical manifestations of the disease, and also their psychosocial and mental health. But we try to look at it in terms of the therapies because it helps us, anchor ourselves in the R&D pathway here and know exactly how do we prioritize, what do we fund, where can our dollars be gap filling and accelerate. So there are devices that we have been obviously supporting for a long time and been very successful in, but we also firmly believe that like most chronic diseases, most chronic non-communicable diseases, type 1 diabetes should be treated with a polypharmacology approach here. We do blood pressure treatment that way, lipid disorders that way, bone disorders that way. Why not type 1 diabetes? Of course, type 2 diabetes also. Why not type 1 diabetes? And that mental transformation has to be not just in this room, but outside the room as well, in everyone's mind here, because insulin is not the only broken hormone in people with type 1 diabetes. Now we are beginning to see lots of benefits, which I'll speak about in a minute, of these therapies. And then, of course, the person that is experiencing the disease and how he or she is handling it and what can we do to improve that. Again, we are all proud of the significant advances made in people's lives today. The life expectancy gap is narrowing. It's not zero. There is still a life expectancy gap in people with type 1 compared to people without type 1, and that we think is unacceptable. Complications have reduced. They have not been eliminated because we don't have anything called perfect glucose control. The average A1C in the leading nation in the world, the United States, is far above the ADA recommended guidelines here. So there are still unmet needs here, and there is a lot we can do and we have to do here. Again, the same pattern of slides here, where we are today, you see that it's been insulin, insulin, insulin. And of course, pramlintide, as I mentioned before, has been approved, but not with significant uptick here. We have to thrive for a better future. We have to be visionaries here to get us there. And we and others are supporting significant research in this area. And I'm just hoping that we will have not only the option here, but we'll also have a commercial landscape here that thinks of type 1 diabetes. And I will mention this here. Obviously, the devices have been transformational, especially CGM, but also the closed loop systems, pump use, better pens, smart pens, decision support tools, so on and so forth, have been amazing. I don't think I need to spend time on this and the next slide, which I'll show briefly here, to this crowd here. We're very, very thankful that you all have been champions in the field, actually in the day-to-day, taking care of so many people with type 1 diabetes in a way that was not seen even 10 years ago here. And this slide, no matter how many times I try to make it or we try to make it, it's dated. And that's because of the progress that's been made. So this is still missing a few things. I wanted to fix it, but I ran out of time. I'm sorry about that. But so this is quite a comprehensive slide. We have certainly Helmsley Charitable Trust, NIH, and JDRF, and many others to thank for this collective work here, and certainly the companies and their commitment here to improve devices. So we are doing very well with this landscape here, and there will be further advances that will come along the way here. But I think today people have choice. It's mostly covered, although there are still challenges. There's a huge disparity in access, and we have to fix that. And I know the exchange is doing significant work in that place here. As I said, and I will repeat many times, we have to take an approach beyond just the insulin-centric view here. This is a trial intentionally termed clever here. Actually, it's not what it means. It's basically closed loop combined with verapamil here. You all are very familiar with verapamil, 50-plus-year-old blood pressure lowering medication approved up to the age of 7, even for pediatric use here. we have supported a lot of basic research and one clinical trial that showed that the target of, or at least one of the targets of verapamil happens to be the thyroidoxin reducing protein TXNIP. And that is pretty activated in beta cells in a stress condition. The stress could be metabolic, stress could be something else, but it gets very activated here, and that abrogates beta cell functioning. But if verapamil was used in preclinical models and in a very small clinical trial of about 19 people, adults with new onset T1D, we saw a preservation of C-peptide. And then we repeated the study along with providing good glucose control. As has been known for about 30 years or so, good glucose control is very required. However, it doesn't somehow, the hypothesis of glucotoxicity protecting the beta cells in type 1 diabetes hasn't panned out. So in the top panel, it shows that the good glucose control in the people with hybrid closed loop systems provided very, very good time in range in all the individuals that were treated with closed loop systems in this trial. And we have many of the investigators and sites in the room here. However, it did not protect the beta cells, but verapamil did in a statistically significant way. We have many follow-on studies now ongoing with verapamil and with combinations with verapamil here. What else can we do? I did say to look beyond insulin, but there's a lot more we can and should be doing in the area of insulins here. And again, a little bit of biology here for people that are quite familiar with the biology. Insulin today is certainly not physiologic, right? We're delivering it at a location where it's not made, where it doesn't work under the skin. It's not fast enough. It's obviously not sensing the levels of glucose, and it's not going to the business center in the body, which is the liver. That's where the pancreatic duct dumps almost the entire part of insulin within seconds there. We all know that. How can we get to that physiologic level of insulin? So we have some priorities here in trying to improve the insulin. It's been a very, very challenging field to change the course, to change insulin. And some of it actually is commercial. But again, there are a lot of academic research. There is a lot of academic research going on, and certainly some company commitment in trying to come up with glucose-responsive insulins and other types of insulins here. But I think this is an area where the bar is a little high here to be able to show superiority compared to today's insulins. I see Mr. Panzier in the room, and I hope he doesn't pounce on me now. But what can we do to advance our current hybrid closed loop systems? I just spoke about insulins being non-physiologic, right? So one of the things we can do is the speed of action of insulin or the site of delivery of insulin, so on and so forth. So we have many efforts going on here. To the left is a project we are very proud of. It's still in discovery phases. This started as an innovative grant with only $100,000 funding. Now it's a much bigger project. In corn snail insulins, trying to use some of the sequences in corn snail insulin to put it into human insulin, because guess what? Snail, right? They're snail. They're slow. They're lazy. However, they prey on only fresh fish, but they won't go fishing for their fish. What they do is when they're close to a school of fish, they do something and the fish become immobile, but not dead. They basically inject insulin into the fish. They put them into a hypoglycemic shock. So we're trying to take the sequence from that and put it into humans to see if we can make human insulin faster, not people putting in shock, but can we make it faster here? So anyway, it's a discovery project. It's very promising, and it's one of those disruptive things that, if successful, will actually truly transform insulins there. But again, the bar is very high. There are other ways to close the loop. In the middle of the panel, there's a very old study we had funded using Simlin or pramlintide as the complementary insulin hormone and taking off the prandial peaks there. And you'll see that of course, people were wearing two pumps. This is an old system here. But you can see the benefits there. This was done by Professor Riddle in Oregon here, and Amylin Pharmaceuticals was our partner here. And then, of course, there are significant enhancements already in the algorithm development. There's more that one can do in terms of personalization, in terms of meal challenges, exercise, so on and so forth. And Helmsley, NIH, and us are supporting significant work also in this area. There's another area of interest that at the moment, as far as I know, Helmsley and JDRF are the only funders here. Maybe others are. I'm not aware of this. And there are some companies involved in this, is ketone levels are a problem as much as anything else in type 1 diabetes, but measuring ketones has been challenging. Urine or blood, who gets the strips gets outdated. It's like a glucagon kit. And so can we have the functionality of monitoring ketone levels, but not have another device, but combine that with a CGM? So we have several dogs in the fight here, and hopefully we'll have one or more option coming out soon. Some of these projects are co-funded with the Hampshire Charitable Trust, and some are supported by JDRF here. And we're hoping that this will add not just the capability of managing ketones, but will enhance closed-loop algorithms, will allow the use of adjunctive therapies that bring with them a risk of ketosis, such as SGLT inhibitors. How much time do I have? Can someone tell me? I could go on. No, I'll try to finish in 5 minutes here. So I started my talk today with adjunctive therapies here. Again, if nothing, I want you all to take home this message here today that we need to be looking at type 1 diabetes beyond the way we have been looking at it. And some of you are involved in this here. It is a multi-organ damage in the body, like with most metabolic diseases here. And also with the increase in overweightedness and obesity, it is not helping, right? In the US as well as outside the US. And T1D Exchange has done some excellent work in demonstrating that at an epidemiological level here. Again, this is a way we should be looking, and I'll give you some examples of work we are involved in. Furthermore, again, I don't want to make this too depressing here. These are some stats here that you are all familiar. Actually, a lot of the data came from the T1D Exchange, so I won't belabor you with this, but we have a gap in clinical care, in outcomes, in both clinical outcomes and mental health outcomes here. And there are things we can do to fix. There are things that we have to wait, such as a preventive therapy, such as a cell replacement therapy, or a truly disease-modifying therapy. There are things that we have to wait. The corner is not going to bend tomorrow there. However, there are things we can and should be doing today with the responsibility, and ensure everybody has access to it. Everybody should have access to it because it is our, again, I repeat, our responsibility here. This is a busy slide, but I'll quickly walk you through. There are multiple examples here that we and others are funding, and I can't read from here already, but anyway. So this is an example here which showing the individual benefits of SGLT inhibition. And I know probably everyone in the room is familiar with this, but this particular class of drugs has been called now the new miracle drug in metabolic area. So much so that, is there any cardiologist in the room? No. Hardest people to have change in mind. I came from that field in my graduate school days here. The American Heart Association changed their care guidelines, including SGLT inhibitors as a first line of therapy here. And now the renal people, the KDIGO guidelines are also changing with that. I just came from the ASN meeting that they're talking seriously about changing these guidelines even furthermore. So they are actually a lot more beyond glucometabolic control. As you all are aware, they now have indications in cardiac and renal complications in diabetes and people without diabetes. Guess what? The only exclusion is type 1 diabetes. We got to change that. And then there are other therapies here, GLP-1s, and probably many, many others, finerenone, and several others coming here. For both, well, finerenone is not for glucometabolic control, but that as well as for complications here. And this is just one study that was published recently here. And there are many, many more that we are funding. We're also funding a study along with Helmsley using a glucagon receptor antagonist in combination with SGLT. And so we'll see what those results come out. A lot of the biology from type 2 can be translated to type 1. We're fully aware that you can't translate everything from type 2 to type 1. And those differences will become very important, particularly in the incretin area where the glucagon regulation is very different in type 1 versus in type 2. This is a summary of the area of drugs and devices here. This is our vision of where we should be going in the future, and we are feverishly pursuing this. And to end here in complicated psychosocial, again, my last pattern of slide here, where we are today, we are very thankful to the people in retinopathy here. Thank you, Risa. Thank you, many others that have not excluded type 1 diabetes, and the companies, of course. This is one complication where we are very happy that people with type 1 diabetes can avail the treatments that are coming out for diabetic retinopathy. Although essentially it's one line of therapy now, the anti-VEGF therapies. There are hopefully more that will come addressing the neurodegeneration before the vascular degeneration in the eye, but we don't have an approved therapy there. But again, I repeat for the second or third time, kidney and heart and other complications, type 1 is an exclusion in commercial trials, and we have to change that. Our focus here are retinopathy, cardiomyopathy, and nephropathy here. Not to say that the other complications aren't important, but as I said at the outset, we have to prioritize somewhere. We are, at the end of the day, a not-for-profit funder that we have, resources that are pretty darn good, but it's not unlimited. It's not, and so these are the areas of focus where we think our dollars can make a difference here. For example, filling a gap where a trial has excluded people with type 1 diabetes. And how can we bring that to bear here? We have a lot of clinical work going on, and not a whole lot of preclinical work, a lot of clinical work going on here. I just gave you 3 examples of trials we're involved in. Some of these are pivotal trials. Some of these are academic-run pivotal trials in partnership with pharmaceutical companies, while some others are entirely academic trials. We're hoping to bring them to the FDA. We are working very closely with the various divisions at the FDA and other regulatory agencies, including the cardiorenal division there, and trying to convince them the benefit to risk ratio in people with type 1 is very different from people without type 1 diabetes, and they need to have a different lens in looking at this. And then we are definitely pursuing many other therapies beyond anti-VEGF, which we all know are effective but quite invasive, and not everybody responds to them. And so what are the other options there? And then this has been the theme, where we are today, where we want to be. And that you saw that in many of our slides before, and so also in complications. I'm rushing a little bit here because this is kind of a summary here. And then in psychosocial, this is a very, very big area. And we, again, have got to prioritize. So it can be, your psychosocial challenges can vary on not only your stage of diabetes, but also your duration and how long you've had it here. And so the interventions will vary. This is a slide basically showing that the subclinical stages when we should be intervening, like in any disease area, because until it gets to a clinical stage, but it becomes very, very difficult and sometimes intractable to be able to change the course here. With that said, we have certain areas of priority where we're prioritizing a little more than the others. We can, again, in order to be able to make a difference here. Many trials, it's all on our website. If you're interested to take part in these, be an investigator, be a site, please reach out. I'm happy to share my slides with you all later. And this is another couple things here. We work across the pipeline. We have internal bench strength here at each of the areas. I did not have time today to address our regulatory and health policy and other efforts we have ongoing, but hopefully at some other time we can talk. We also work with as many people as possible around the world here, other not-for-profits, federal funders, commercial sector, so on and so forth here. So thank you for your attention. Happy to take questions now or later.
Thank you so much. Wonderful talk. Carol Levy, New York City, Mount Sinai. So I want to bring up something a little bit different here, but just to bring to everybody's attention. And it relates to what you referred to in terms of specifically closed loop systems, adjunctive therapy, how it can help. I think most people know in this audience that one of my focuses is pregnancy. Mm-hmm. It's a critical area. I want to thank Helmsley and prior work by JDRF for moving that forward. The piece I want to bring up right now, though, is a really important one that we discussed at dinner last night, which is the discontinuation in the US of Levemir. And I realize this is completely out of what you just presented, but the reason I'm bringing this up now is because I think there's an opportunity here for us to advocate. And I'm saying this now with you and David here, because loss of this insulin, for those of us who are old, who remember the days of discontinuation of Ultralente and other ones, but this is one of the few insulins that is really beneficial in pregnancy for those patients who refuse to use a pump. If we can get something together for that, I think it would be important. Second thing is the SGLT2 inhibitors are great. We have to have the ketone piece. For any of us who've been in the audience, who've seen someone present in the ER when their catheter blocks and their pump, we can't keep having that risk.
So, I couldn't agree with you more on both counts. The first one we'll take back and address. I will be honest, we haven't addressed the first point you raised here. We at JDRF haven't addressed it, but we will definitely go back and discuss. We'll also discuss with David's team and see what we can do collectively here. The ketone meter, I couldn't agree more with you. There are ways we can still use them carefully. If you look at real world evidence from the DPV and other places.
I totally agree with you.
Yeah, exactly. So there are ways we can do for patching the gap until a CKM or a CGM, CKM becomes available. That will take some time. Abbott is obviously leading, blazing the trail here, but we're hoping there'll be other, just like in CGMs, other competitors come along here.
Yeah. And then the final comment is rollout. So great work on TZL, phenomenal. The rollout has been a little bit tricky. And I wonder if we need to think more as a group about how to collaborate on that. You guys did the study, it's great, it's wonderful, but then we're all of a sudden like, what do? We depend upon industry, but industry doesn't always have that medical piece that we have.
Trust me, Carol, we are all over it.
Okay.
all over it.
Very happy.
What is our role, what we can do here? Osagi. Oh, sorry.
Thank you. Thank you so much. It was a great talk. I'm Mike Natter. I'm one of the endocrinologists here at NYU. I was curious about the verapamil and if you saw a lot of hypotension while folks were on verapamil at the same time.
Actually not. And we have two of the site investigators, at least two that are in front of me here. They were monitored very carefully and closely. Of course, it's a clinical trial. And this was especially an intensively monitored clinical trial. In the real world setting, when this is used, we have to be very careful. But in the prior study, which was not in pediatrics, and this study, which was in peds, we didn't see any major thing. I think there was one event, and that was also corrected. It wasn't a serious adverse event. So nothing drug-related hypotension. And the doses were very carefully chosen. And it wasn't given, I won't get into the details, it's all in the paper there about body weight related dosing that was done here. And I think 30 kilogram was the lowest cutoff for people being eligible to be on verapamil. These were 7 to 17-year-olds. So. All right, thank you all. I'll be around if I'm happy to take other questions.